Welcome to the third episode of Current Affairs series for Civil Services Mains exam. Today, we're discussing Garima, India's first gene-edited sheep, a major milestone in agricultural biotechnology. At the end of this episode, we will be telling you what could be model question based on this topic. For the model answer, you can contact us. Dr. Sharma, could you explain how Garima was created and why this is such a landmark event for Indian science?
Absolutely. Garima was developed at the National Dairy Research Institute using CRISPR-Cas9 gene editing technology. Essentially, CRISPR works like molecular scissors, allowing scientists to precisely modify specific sections of DNA. This precision surpasses traditional breeding, which is often like rolling genetic dice.
That's a great analogy. How does this precision translate to practical results, especially compared to conventional selective breeding methods used in Indian livestock farming?
Traditional breeding can take generations to isolate traits, and it's often hit or miss. With gene editing, we can target, say, a single gene responsible for disease resistance and alter it directly. In Garima's case, this means faster and more reliable development of desired traits.
Before we move on, could you briefly describe CRISPR-Cas9 for listeners who might not be familiar with genetic engineering tools?
Of course, CRISPR-Cas9 is a protein complex found in bacteria, originally used to defend against viruses. Scientists repurposed it to cut and rewrite DNA in animals and plants. Its accuracy and efficiency have revolutionized genetic research globally.
Now, Garima's development focused on disease resistance, particularly to Jones disease, which plagues many herds. Dr. Sharma, why is combating diseases like Jones so vital for Indian farmers?
Jones disease, or paratuberculosis, severely affects sheep and cattle, leading to weight loss, decreased productivity, and often death. A 2020 study estimated Indian livestock losses at over 500 million annually due to this single disease.
That's staggering. How does gene editing improve resistance, and is this approach more sustainable than traditional vaccination or antibiotic use?
By tweaking immunity-related genes, we can make animals innately resistant, so they're less likely to get sick in the first place. This reduces the need for antibiotics, which is crucial since antibiotic overuse is driving resistant bacteria, a global health crisis.
So, in effect, gene editing could reduce both disease outbreaks and the risk of antibiotic residues in our food supply?
Exactly. It's a win-win for farmers' lower veterinary bills and healthier animals, and for consumers who get safer, cleaner products. Plus, it's more environmentally sustainable since fewer chemicals enter ecosystems.
Let's dive deeper into the comparison between gene editing and traditional breeding. What are the main limitations of conventional methods, especially when aiming for traits like higher milk output or better wool?
Traditional breeding is slow, sometimes requiring decades to combine desirable traits. It's also imprecise, as unwanted traits can hitchhike along. Gene editing, by contrast, lets us target only the genes we want, much like editing a specific word in a manuscript.
That's an insightful analogy. Are there examples from other countries where gene editing has already shown tangible improvements in livestock?
Yes. For instance, in New Zealand, gene-edited cows have been developed to resist tuberculosis, and in the US, pigs have been edited for enhanced disease resistance and leaner meat. These examples demonstrate the global momentum of this technology.
Given these successes, how might Garima's birth accelerate similar advancements in India's livestock sector?
Garima sets a precedent, showing Indian scientists and policymakers that advanced genetic interventions are feasible here. This could attract investment, foster public-private partnerships, and drive further research into traits like drought tolerance or enhanced nutrition.
Before we wrap up, we should consider the ethical and societal aspects. Gene editing in animals is often controversial. What are some of the main ethical concerns, and how is India addressing them?
Ethical debates focus on animal welfare, long-term ecological impacts, and food safety. Some worry about unintended genetic effects or the potential for corporate monopolies on genetic resources. Indian regulators are now developing frameworks to ensure transparency and public engagement.
That's important. Do you think the benefits like improved food security and economic gains outweigh these concerns, or should India proceed with more caution?
It's a balancing act. While gene editing can boost productivity and reduce poverty, it must be done responsibly. Public education, rigorous safety trials, and inclusive policymaking are key. Countries like Brazil have found success by involving farmers in decision-making processes.
As Garima's story unfolds, what's your vision for the future of gene-edited livestock in India?
I see gene-edited livestock as a cornerstone of sustainable agriculture, producing more with fewer resources, while safeguarding animal welfare and consumer trust. India can lead by example if it combines innovation with strong ethical oversight. And finally, this can be model question for Civil Services Mains exam based on this topic. Discuss the significance of India's recent achievement in developing the first gene-edited sheep, highlighting the potential benefits and associated ethical concerns of such biotechnological advancements in livestock.